Good morning, good afternoon, and good evening. We are on the 293rd session of continuous Zoom session of International Forum for Promoting Homeopathy. So before we proceed, I would like to have 1 minute silent prayer for the success of the vision and missions of IFPH and also for the successful conveyance of 2 successive meetings, one after another, one in English and another one in local language, Malayalam. So please let us have 1 minute silent prayer. Thank you. So today we are on the 293rd session, continuously conducting and uninterruptedly conducting. And we are on the same process of educating humanity at large regarding the potentials, possibilities, and effectiveness of homeopathic medicine in all segments of life, plants, animals, and human beings. So our motto is one world, one health, win with homeopathy. With that motto, we are marching towards a unique session on agro-homeopathy, and our chief guest today is Dr.Ileana Rindasu from Romania, who is a medical doctor specialized in homeopathy, rehabilitation, and physiotherapy, General Secretary of Romanian Society of Homeopathy from 2007 to 2018, and President of Romanian Society of Homeopathy from 2018 onwards. Vice President of Commission of Alternative Medicine from Ministry of Health, Romania from 2018, and General Secretary of European Committee for Homeopathy from November 2020. Professional experience, Dr. Rindasu is lecturer of homeopathy at the National School of Public Health, Management and Continuous Learning for medical doctors, for doctors who obtain additional qualification in homeopathy in Romania from 1995 till present time. Responsible for regional homeopathic teaching in Bucharest at the National School of Public Health, Management and Continuous Learning for medical doctors from 2015 onwards, and member of LMHI, Liga Medicorum Homeopathica Internationalis in the Education Committee 2014 till present time. Dr. has diploma in competence in acupuncture, diploma of competence in homeopathy, and a PhD student at Bucharest, the capital of Romania, Faculty of Horticulture, pest control through agro-homeopathy in fruit species. So the topic of discussion today is agro-homeopathy, an interesting topic. We have a very important patron of homeopathy, Dr. Latif Sir is with us, who is also our chief person dealing with agro-homeopathy on behalf of IFPH. We will be conducting further interactions with Dr. Abdul Latif Sir also. Over to you, Dr. Ileana Rindasu, for your session. We need to wind up exactly in time at 9:30 Indian time, because we have another session in our local language with the political leaders from Kerala. So you can start, and over to you, Doctor.
Yes, thank you. So I have half an hour, or how much?
You can take up to 40 minutes, 30 to 40 minutes.
Okay, thank you very much. Good afternoon, good evening, or good morning to everybody. I'm very happy to be here with you, and thank you again for the invitation. I want to present you today, as we spoke, my work on the thesis that I finished last year, PhD, at the Faculty of Agronomy, actually Horticulture from Bucharest. And there I found very open-minded people who accepted me to work with homeopathic remedies and to show that they have an effect on plants. They were very curious to see if this was so or not. And luckily, I found very good methods to prove the value of homeopathic treatments also in horticulture. I would like to share the screen if I can, if you allow me to share screen.
Yeah, please proceed.
Let me see if I can.
Yes, you can.
One participant can share at a time.
Yeah, you can.
Okay. So I made a PowerPoint. This is actually the research I made, and here you have, let me see if we can make a slideshow. So this is the thesis. You see the exact title is Pest Control through Agrohomeopathy in Fruit Species. The work consists of 8 chapters, 22% is the general part, the rest are personal contribution, 132 bibliographic works, and the capitalization of the results was achieved by publishing 7 scientific articles as first author, out of which 3 articles listed ESI. The research was performed during 3 years, from 2017 till 2020, and with the aim to study the effects of homeopathic preparations on pest control in fruit species, to see also the changes the homeopathic remedies produce in plant physiology. The first part was a bibliographic study, as usually is made in such situation. When you make the thesis, you have to study what is the situation of the research made before your own research. In chapter 1, I emphasize the importance of homeopathy in the context of current scientific research, underlying that in recent years, from what I could see, homeopathy is very much perceived as a nanomedicine or even a nanopharmacology. A Romanian and professor and academician, Dumitru Dobrescu, has written this in his book, which seems to be the first ecologic homeopathic pharmacology in the world. He wrote about the principles of action in pharmacology of homeopathic medicines, and he says that homeopathy actually is a nanopharmacology, and this opens the road to be recognized by other researchers which are not homeopaths, but to have a common language with them and to be able to somehow answer to the denigrations which were made many times during the history of homeopathy. Now we have this tool to show them that there is a nanoscience, and actually from the centesimal dilution C6 or the 50 millesimal LM1, nanoparticles can be found in the elements from the original substance, as Professor Rajendran is explaining in his works. I put in my thesis also some pictures from his book Nanodynamics. You can see the nanoparticles photos from electronic microscopy. I chose here Natrium muriaticum LM30 in certain scales, 100 nanometers or 10 nanometers. Also from CH6, as I said, also we can find nanoparticles. You can see some photos at the 2 scales, 2 different scales. In chapter 2 till 4, I presented also some general aspects, different agrohomeopathic studies in pest control and phytopathology, as well as the effects obtained on growth of healthy or biotically or abiotically stressed plants. There is a very rich bibliography in this domain. Chapter 3, I looked for the effects of homeopathic medicines on germination and plant growth, and I found that the isopathic model is the most commonly used in such studies. And what is the isopathic model? Maybe you know already. It's a substance that initially is used in high doses as a stressor, which later on is used in homeopathic doses for study or treatment. And this is considered an isopathic treatment, and it's very successful in agrohomeopathy many times, such a model, for research at least. In chapter 4, I presented a synthesis of previous work on agrohomeopathy with the role of different types of homeopathic dilutions, seen as low or high dilutions. And it's interesting that usually low dilutions were used, even decimals or centesimals, but not higher than C12. But other researchers later on started to use higher dilutions. And also I use high dilutions because I was convinced they are acting also. Now, in the second part of the research, I presented the purpose and objectives of my own experiences. And these objectives were to see, first of all, the effects. I thought that it's easier to find out about the effects on plant germination because there is already a model of research to be made in plant germination with homeopathic remedies. And I used an original model with Asimina triloba dunal, which is the North banana or pau pau called. And I use such seeds to see if homeopathic preparations act on these seeds, on their germination and early growth. And then I started to see the effects of homeopathic preparation in the control of pests. And the professor with whom I worked at the University of Agronomy from Bucharest, Professor Stanica, he proposed to me to see the effects on Acheria cuco, which is a mite that attacks goji berry plants. And also I worked on Eriosoma lanigerum attack on apple trees. And we together saw the effects of homeopathic preparations. Now, the first experiment, as I said, concerned germination. I used the Asimina triloba model. And I made two studies. First was a preliminary study in 2018 from January 23 to 15 of April. And then I made some improvements, repeating the same type of study in 2019. I will show you what kind of improvements I made. As I told you, Asimina triloba is the northern banana, and at the faculty from Bucharest, they made different type of studies on Asimina triloba. They appreciate very much this model of study in other domains, not only homeopathy. They worked since 1995. Here I show you the germination trays that I used in the first year, and you can see the variants. There are 8 types of treatments that I made, actually 7 plus the control group. I had 3 trays where I used 3 dilutions of natrium muriaticum from decimal 6. Then I passed to the CH30 and CH200. I also used the same time of dilutions with another remedy, silica. And I used also for one tray, it was a curiosity of mine to see if diluted hormone, a homeopathic dilution of hormone can have good effects on plants. And this is the growth hormone releasing factor that I used in CH30 potency. And so this was the study I performed first in 2018. In 2019, you can see other types of trays. They are alveolar trays, and here I could see more precisely the number of seeds that I use, because in the first type of study, I was not thinking exactly of the number of the seeds, but it turned out that it's very important to know the number. So I used this type of trays in the second year. And I recorded. Here is maybe a little bit boring, but I tell you exactly what I did, and I will be more quickly passing to some details which are more interesting. So you can see I had to know the number of germinated seeds and the maximum length of the stems. Also, I measured the length of the roots, and later on in 2019, I could calculate, knowing precisely the number of seeds, the mean germination percentage, then an index which is quoted by Patil and Delian, seedling vigor index. It has a certain formula of calculation. And in the second year also I calculated the fresh and dry mass of stems and the fresh and dry mass of roots and the amount of chlorophyll and carotenoids. Yes, then the length of the stems and roots, the number of leaves and their areas. All kind of details were important to show them what are the results. Here I showed you, I wrote about the method I used using a spectrophotometer and the extraction of the pigments in 80% acetone for the amount of chlorophyll and carotenoids. Here is the statistical analysis, what program was used, and the statistical significance was considered as usually for a p lower than 0.05. Now, let's see some results quickly, not showing every detail. In 2018, for instance, what I could observe was that the seeds treated with silica CH200 germinated later. But even if they germinated later and in a smallest number, they had a very good growth after the germination began. And with Silicea CH30, we obtained the largest number of germinated seeds in all cases and in all trays and repetitions. Then the next values were of Natrium muriaticum, where CH200 had better results than CH30. Also, the dilution of the hormone that I used led to the highest number of germinated seeds in the beginning, but then in the end of the study, it was not maintained this advantage. So in the beginning, there was the highest number of germinated seeds, but in the end, this was not maintained. The Spearman rank correlation is this means the statistical analysis showed differences which were significant, so statistically significant. Here I show you, for instance, the tray with seedlings of Asimina triloba in the end of the study in 2018. We can see there were not many seeds in the end of the study, but they had the highest growth from all trays. This was important. And the highest growth after this hormone we obtained, as I said, with Silicea CH200, and this was also statistically significant. Here I show in a graphic the influence in the next year, in 2019. It's very interesting that if we take the moment when I consider that the study of the germination is finished, and then we had to study the early growth after 11 days. So in the first 11 days, what was the result? As we see, the control group is here. In the beginning, there was no growth or no germination in the control group, but in the other groups, already germination began and it was important. And the highest germination was obtained again with silica, but in a low dilution. This decimal 6 was the most important here. As you see, the biggest value. Again, the statistical analysis shows important differences between the variants. And for instance, I gave here as an example the photo with silica decimal 6, which was the best in germination. You can see how many seeds germinated and the seedlings were already grown here. The seedling vigor index also shows very interesting details in research, and the highest value was in this silica D6 treatments. At the end of the study, as I said, I also had to measure the roots, the mass of the fresh and dried roots, and also we study the fresh and dried mass of the stems. And we see here differences between them, with the highest mass of fresh and dry roots obtained with the dilution of the hormone. Here is the highest value. And you see the tray treated with the dilution of the hormone. It's difficult to perceive on the picture, but they are the highest, the most well-developed, yes, seedlings in the same time as we compare with the other trays. About the root length, you can see that the best results were obtained with the Natrium muriaticum CH200, followed again by silica in this decimal 6. For stem length, all variants differed significantly from the control group. This is interesting, yes, that all variants, with the except of the first two, they were significant. And here we have the tray, the picture of the tray with the control group. So we see, if of course we had also the counting made, but you can see also from the photo, there are very few germinated seeds, yes, and the height of the plants is not so much, they are not so developed. Some leaves indeed are have a big surface, but only few of them. About the assimilative pigments, I made, as I said, the study, and the highest amount of chlorophyll was recorded with, again, with this GHRF CH30. The highest amount of carotenoids was obtained in another group, Natrium muriaticum D6. Here we have the graphic form. So in conclusion, in 2019, when the study was finished, Silicea D6 proved to be consistently useful for obtaining a high germination percentage and a high vigor, the effects being maximum at the end of the study, also on the length of the roots. Here we have a slight difference from 2018, where the best growth was recorded with Silicea CH200, and this leads to a kind of discussion why there is a variation of the dilutions from one year to another, and this was not happening only in my study, it happened also in other situations, in other studies performed by researchers before this one. And this is a team for research, for other researchers maybe later on, to see why the dilutions are not exactly the same, which have the same result, but we can see here that the silica preparations had the more significant effects compared to the other remedy, which was the Natrium muriaticum. And the hormone, of course, the dilution of GHRF had also an important effect, yes, and especially on the amount of chlorophyll and on the mass of fresh and dry roots. This was the first experiment which tried to show that the physiology of plants is modified and that there is something happening with the chlorophyll, with the carotenoids, and maybe with the enzymes and other substances which I did not measure in this study, but there is a modification of the physiology of the plants, not only, I mean, here is an effect which has to be, of course, more detailed in future, but it has to do with the effects of the treatment on the pests, because we do not treat so much, we do not destroy the pests so much, but we work on the plant physiology and we make the plant more resistant to the attack of pests and other negative elements, maybe microbes or fungi or some other external noxa. Now, about the second experiment, here I already started the study on the pest, on Acheria puco, on goji berry plants, Silicium barbarum. The study was made using lithium leaves attacked by Acheria puco according to the principle of similia similibus curantur. In fact, I can consider this like a nozzle. So I tried a very simple way of treating with a kind of a nozzle and see if this functions, and it indeed had results. But also I used the Tuya CH30, knowing about Tuya that it has immunomodulatory effects in humans. So I thought that it can have a very good effect also on plants. And also I had good results with Tuya. The experiment was taking place at the vegetation house of the faculty. You can see here the pots which I used. The treatment started at the end of 2017, at the end of the vegetation period, trying to prepare the plants for the next period of vegetation, and then again in 2018 and 2019. The biological material, 2 groups of 10 pots of 2 genotypes. This is what the faculty proposed, to use 2 genotypes, which they called B1 and B2. And so I had the first variant was the control group, then the second, a chemical control, where a chemical substance was used, abamectin. Then I used the Acheria Cuco remedy, CH6. Then I had also CH30. And I bought from the pharmacy a homeopathic remedy, Tuya CH30, which I used in the study. The Acheria Cuco dilutions I prepared myself, starting from the raw material. Then we made a maceration of leaves attacked by the pest. You can see here a vial where I made and I put the maceration, and it was put in alcohol, and afterwards I made the dilutions using the Hahnemannian method. So I prepared myself with the old Hahnemannian method. Then I studied with this treatment the frequency and intensity of the attack, the length of the stems, and the degree of fruiting of the goji berry plants. Now here there are some very detailed results. Maybe it's not worth reading them all. There were different results in 2018 on genotype B1 and B2. It turned out that Tuya CH30 had very good results all the time. And in the beginning, good results were also obtained with Acheria CH30 in B1. Then in the second month, because I made this determination on longer periods of time, the situation changed a little bit, and we see here that mainly Tuya CH30 was obtaining the best results, but Acheria CH30 also maintained very good values. So there was, for a moment, I mean, in the beginning, there was some determination very good in favor of CH6, Acheria Cuco, but then Acheria CH30 turned out to be better. In the next year, at the beginning of the season, again, of the vegetation season, after a period where no new treatments were made, the effects of the treatment from previous year showed out to be still present, while the other plants from the vegetation house had signs of attack. The plants treated in the previous year with Acheria Cuco dilutions and with Tuya were like this, as you see in this photo. So they were having very good leaves, normal leaves with no galls. There are some, you can see on the leaves usually this type of galls made by the mite, by Acheria Cuco. So here nothing is present. The evolution of medium intensity of attack in genotypes B1 we can have here. And we see that, again, Tuya CH30 is the best. Tuya has a very small attack compared to the control group, and also compared to the chemical control even. Yes, and also Acheria CH30 has a good result. Here we have the graphic situation of the medium intensity of attack. So we have again the control, and we have here Tuya in a very good position. And also Acheria CH30 is good comparatively to the control group. What we can say regarding the medium frequency of the attack, that positive results were obtained in both cases. In relation to the untreated control, and we had statistically significant results for Tuya CH30. Also, this remedy showed the best results on the stem length and on the number of fruits that were obtained. Here we have the graphic of the medium frequency on B1 and B2. And as a conclusion, homeopathic preparation used, we concluded that they contributed and can contribute in future to the health of the goji berry plants as an accessible measure for farmers, taking into consideration that Tuya CH30 and mainly Acheria CH30 dilutions showed favorable results in controlling the attack of Acheria pupo. So in the end, you see what beautiful leaves these plants have, no gores on them. And when we counted the number of fruits, the number of fruits was best in, as I said, in the treatments with Tuya CH30 and also some results with CH30 Acheria pupo. And I hope I have time to explain a little bit also about the second and last experiment, which regards the attack of Eriosoma lanigerum on the apple trees, Malus domestica. I had here a sequence of experiments. So one of them was performed in the laboratory of the faculty, actually of the research center connected with the faculty. We put some branches attacked by Eriosoma lanigerum on Petri dishes, and they were treated with remedies. And after 72 hours, I observed the pests on the Petri dishes, and all of them were dead, immobile, except for one dish where there was a viable insect. But it's what's interesting that this insect did not have the waxy filaments that usually these insects have, and they contribute to the protection of the insect in front of the attacks from outside. And so I think that the homeopathic treatment can also have an effect on the pest directly. So my hypothesis was that the effect was mainly on the plant, but here also on the insect. For those who know how this insect looks, it has waxy filaments, and here they disappear. This is very interesting when we regard this insect at microscope. Another study I could do also to see the effect on Eriosoma lanigerum was a study in the laboratory again, and one part of the study was in the greenhouse. I used some seedlings in pots, and I made an infestation from affected apples from the orchard or the faculty. And so we infested artificially these seedlings from pots and made 3 groups to study. It was a small study, just a preliminary one with 2 pots in each group. V1A and V1B, I used remedy Citrus paradisi because I read about Citrus paradisi. It has a good effect on other pests, and I wanted to check the effect on Eriosoma lanigerum. And also I made the remedy, again from a maceration made by myself from the body of the pest, Eriosoma lanigerum, and I used the potency CH30. Also, we had the control group on other 2 pots. Here you can see the pot, which was a healthy seedling, but near it, we put a fragment of a seedling from the orchard, which has this infestation with Eriosoma lanigerum. And here I describe what kind of rhythm of the treatment I used and what were the quantities, yes, 100 milliliter of solution for each pot and so on. And we made the comparisons of the number of shoots and the number of leaves and the total leaf areas. At the end of the study, the best effect was obtained with Eriosoma lanigerum, but some results also were with Citrus paradisi. And this encouraged me to continue the study going to the orchard to make treatments on bigger areas. Here you can see the pots. So good results were obtained also with Citrus paradisi in this first group. But the best I consider when I consider the number of leaves and their areas the best result was with Eriosoma lanigerum. And here is the control group, completely destroyed, although it was correctly watered like the other groups too, but because of the pests, these seedlings did not survive. Now, when I went to the orchard to see the effect there, the experiment was already began in 2018 with biopreparations used for 2 months in August and September, a period that overlapped with the second annual maximum of this pest woolly lice population, or Eriosoma lanigerum. Then I continued to treat the trees in the next year between 1st of June and 1st of September. And I treated 6 rows of organic apple trees. Here we have in the image seen from above, the rows from 41 to 46, and with varieties called Topaz, Gold Rush, and Rubinola. Here I explain exactly what I did. I took as method of study the measurements of the pest colonies from the base of the trunk, because I had to compare some data, and I saw that the pest colonies were developed on a certain area on the base of the trunk. So I started to measure this and to make comparisons of the affected trunk area for each row between 2018 and 2019, and I used for statistics the exact Fisher test. Here we have the results. I think it's not worth to read all of them. I will tell you just the conclusion. In 2019, because it was very difficult to quantify very well so many trees, there were about 42 trees in each row. So it was rather difficult to make a very objective analysis. So I used only the first 6 attacked trees from each row, which were marked with letters A to F, and additional treatments were applied to each of the 6 trees by brushing and then watering with 5 liters of homeopathic solution these trees that were marked. Yes, and we have also control rows again with these 6 trees. The second period of study, we have here the statistical comparisons between the rows. The most important is to see together all the varieties, because we have 3 varieties, and it turned out that Topaz was responding much better to the treatment. But the other varieties also responded well, and when they take them all together, we see a favorable effect overall, so that the average frequency of attack was, in the end of the study, 24.19% for the treated trees compared to 51.08% for the untreated or control group. So there is a very important difference, twice as high in the untreated group. Now, another comparison is made for the intensity of the attack, and we have here first in August the measurements made in August 2018, then in June 2019, when I began in the second year. There are some, again, differences between the varieties. And again, I want to show you the overall result. If we take into account the total number of apple trees affected at the beginning and the end of the experiment, regardless of the variety, for the average intensity of the attack, from a statistical point of view, the percentage differences were significant. As we see, the treated group is the first one. There is a very small value, 11%, 0.52 in the end of the study. And here there is a growth from 47 to 86. So there is clearly an important difference in the medium intensity of the attack. Here is what I want to show you that I measured on the trunk. So we have, for instance, a Rubinola trunk attacked by Eriosoma lanigerum in the end of the study in the control group. So we see here there is an area of colonies which is measured and shows certain values. And there is still an attack on this trunk. And here we see Rubinola trunk attacked initially by Eriosoma. In the end of the study, there is no development of colonies on the trunk of the treated tree. So what are the conclusions? Here we see the rows, one of the rows of trees that I treated. In both years, the best results in medium frequency and medium intensity of attack were recorded in topaz, but overall good results were obtained also in the other varieties. And we can see that there is an effect, a significant effect, as I said, on all the trees. The frequency of pest attack remained at lower values in the year following homeopathic treatments after the first peak development of the woolly aphid population, although in the second year we did not perform treatments until June. So again, in the apple trees, like in goji berry plants, we can see that in the next year after the homeopathic treatment, there is still an effect which is preserved from the previous year. This is very interesting and very important for farmers. So there is a direct effect on plant organisms, and they acquire a lasting ability to defend against biotic stress from one year to another. The final conclusions were that we obtained positive results regarding germination and plant growth in Asimina triloba dunal model, which confirms the data from literature, and also another study that I performed with a professor of physiology of plants from the University of Agronomy. We made this study on germination of the of other plants, of wheat actually, and also there were very good results on wheat germination with homeopathic remedies. So these studies prove that there is a direct effect of homeopathic preparations on plant physiology, an effect that subsequently promotes in the mature plant a better defense against biotic stress. Then we obtained positive results in controlling pests such as Eriosoma lanigerum with homeopathy, as observed in experiments that I performed, especially if homeopathic solutions from leaves attacked by pests are used, or the preparations from the body of the pest. Also I obtained very good results with different homeopathic remedy, not a nozzle. I used Tuya CH30 and there were very convincing results. Further studies, of course, are needed to clarify the indication of appropriate dilutions and to be able to make recommendations exactly of which dilution to use for the farmers. Yes. And the problem is that there is a relatively high degree of variability in the response to different dilutions, and it is necessary to insist on knowing which are the factors that influence the response of plants to different dilutions, and to be able to control these factors to obtain stable results. Because if you want a farmer to use your preparations, they need to know exactly what to do and to have stable results. And here I put also the published papers connected with thesis and the research. They can be found, almost all of them, on ResearchGate. Actually, I saw there was big interest on ResearchGate, about 10,000 readings of the general part of the thesis, where I made a review of all the research made until now. And this was very interesting for many because they read in such a great number. So here are all the papers. And this is all that I wanted to present you. And what I want to say is that with this thesis, although I am not an agronomist, and for me, it was difficult to actually to work in a field about which I didn't know very much, but I had the support of the professor who coordinated me, and he gave me some advices what to do about the agronomic knowledge to be able to deal with it. And I did only the part of a homeopath, thinking of what remedies should be used. And also I performed myself the treatments. It was rather difficult. For this reason, I used only 6 trees in the end of the study on each row because I had to go myself, and actually even my family helped me to work in the orchard and to be able to make the treatments. Usually in such research, I think you need a team of people, and when you work at the PhD thesis, you don't have a team, you are somehow alone with your coordinator. But if there is to be done a research, it's clear that you have to have a team of people who are committed to the same goal. And in this way, you can make very good treatments, and I'm sure that there can be very good results. This is what I wanted to show you about, not about agrohomeopathy in general, but about what I did myself, what kind of study I could perform. And I was very happy that people from the university, um, so say, medium, they were so open. They wanted to know the truth, and they were really very happy. And if they saw there were results, they recognized them, and they even want to use this in future. They think how to use these results and to continue the studies. So this is it. I hope you enjoyed my
Thank you.
presentation. Yeah.
Thank you, Dr. Rindasu. So it is good to listen to you. We have another important person with us, that is none other than our patron, Dr. Abdul Latif sir, who is the retired principal of Government Homeopathic Medical College in Trivandrum and the controlling officer of homeopathic education in Kerala. Now retired, Dr. Latif sir is very much involved in agro-homeopathy and conducting researches. Latif sir, please say a few words about this research study from your perspective.
Thank you, Doctor, for welcoming me to the session. I also welcome Dr. Ileana from Romania, and I think she has colorfully set up certain experimentation with due regards to homeopathy in agro-homeopathy, and regarding what is in homeopathy, is a placebo effect or not, and the doctor has clearly established it is nothing but the effect, the merit of homeopathy, other than any, but it did not even use any pesticide or any biomanure water maybe, but this is an eco-friendly method of cultivation. It's better to say purely organic cultivation. But I am searching behind the organic, that really speaking, organic products contains heavy metals, not the pesticides. So we are concerned has to be acquainted or to be recorded on this baseline to see every best product available in the world, present time being organic, but with the accumulated heavy metals is a drastic health concern to human being and all the living or one health. So I appreciate you, madam. Actually, you are one among the expert group uh, regard to the link of homeopathy with agriculture. And even though others are having some concern regarding but this minutest quantity, how this is affected, it is a miracle. Even by by appreciation even there, but they are not at all accepting and freely talking in a mass media or even writing article. But better to see on the reference, we have more than 600 references, even by single symbol inquiry, HOMOEOPATHY or agrohomeopathy. Minimum 600 inquiries with the best possible study with the significancy and with the control or and the best study. But in my case, usually it is better to say from lab to farm. But in my case, it is just reverse. Even if you are having what is a best product with a certified product, water may be with a star value, what may be, and if it has been used to the public and public is not aware about and is not satisfied, what is the importance of? So my homework has started about 6 years time, and finally at the end of 6 years, exactly on 26th uh, January, January 1999, started the experimentation in 1000 yellow leaf disease attacked this arecanut. Within 3 months, the entire condition was reversed back to normalcy, and the control 500 arecanut trees also we controlled back to normalcy. And this was the first experimentation conducted extensively. And from that day onwards, I am also in a busy government job, being the principal of a college with so many, say, what is in an institution, and moreover the staff and everybody and even the public and it is a difference of opinion with the government at the time the controlling of 16 major epidemics at the time, uh, purely about 6 years time. That is very crucial in the control of epidemics at the time. I can't find enough time to concentrate my experimental study with the control. But happily to say, one MSc botany student from the University of Calicut, that is near university, has brought as a effective study with mustard plants, and the main ingredient that is used is PEG, polyethyl glycol. She has applied this medicine chemical compound PEG, polyethyl glycol. That's nowadays it is a content of the vaccine, and so many side effects are there because of the PEG. And that medicine was applied along with and without. And colorfully, she has observed that the chlorophyll content in the 132 percentage increase of protein in the seed that is obtained after about there is an experimental study about the course study that she has conducted, and good examples was given. Similar studies were now started in the university, agriculture university, but they are demanding 4 lakhs per 3 to 6 months study, and earlier they have demanded the entire content shall be submitted, and from that content they have to purchase the medicine from the open market. And uh, I'm having great concern regarding when the result will come at the, or even my death maybe. So it's a condition. It's a continuous. It's a toxic study, and every study is going on. But even then, I am fully satisfied with the saying of the farmers. They are fully satisfied even with the viral diseases or any condition with the climate change, and I had an opportunity to grow 5,200 plantain, banana plantain, in an area where the temperature was 39 to 40 degree. For the last 3 months, not a single drop of water, not a, not a rain, but the nearby river is full of water, but it contains the pesticides. So I scared about, as you see, and with monthly application of about some the stage being 50 ml to 350 ml water once in a month. That is very strange, and no problem, and a good yield about 14 kg per plantain was observed. The taste is so, what I can't say what is the exact taste, like we are chewing this our sweets. And again the shelf life also increased. The problem is that uh, we can have the same thing, the raw thing, and we can travel around the world twice, and even then it may not have any negative result. So tomato was preserved for about 4 months and 10 days without any damage. So the result goes like way, and anyway, the farmers are benefited. Luckily, even with the viral diseases. Okay, sir. Okay, in another occasion, I have enough time to
Thank you,
discuss
sir. Thank
better.
you, sir. Actually, we have one more scientist from Cuba who presented in our platform for agri-homeopathy. I think we will be arranging one more session with the three of you, Dr. Latif sir, Dr. Ileana, and Dr. from Cuba, for another discussion session on agro-homeopathy. I think I will keep in touch with all of you so that we can arrange, according to your convenience, a common day where we can discuss further about homeopathy and in agriculture. That will be very needed for world today, especially at the time of huge pesticides are used, which is having too much impact on the soil, on the plants and animals, and even to human beings. Everybody are affected by that. So we can contribute a lot to bring healthy vegetables for our consumption and also healthy life among the plants. So we need to wind up today. We have 3 more minutes remaining before we start the next session. If anybody wish to ask or talk 1 minute or 2 minutes, they can have time. And I request, Lokmani Gupta sir, you have to speak 1 minute only because we need to wind up, please. Just 1 minute.
Yes. Thanks a lot for the nice session. The 3 medicines are used in agro-homeopathy, and I got very good results with abrotenum, with phosphorus, and thuza. Thuza I used in the trees with nodular boas. Boas with a very small fruit. Abrotenum I used with good results. Berries, amla berries were falling from the tree. So after use of Calcaria frost, biochemic in roots, and abrotenum in roots, berries were standing, amla berries were on the tree, and phosphorus I used to sprinkle in plants for dried leaves of these plants. This is what I want to share. A very good chapter.
Okay, sir. Thank you, sir. So it's your time is limited. 2 more persons raise the hand. Harit Bhagavan, sir, please.
Thank you. Thank you. I have a lot of questions on this topic, but due to lack of time, I am not going for that. I am reserving my discussion on the day.
We will.
Definitely.
We will have one more discussion time for this.
Definitely. I am waiting for that. Thank you. Thank you very much.
Okay, thank you. Dr. Sridhara Kurup, sir, please. Just 1 minute only, sir. Sridhara Kurup, sir.
Okay. I congratulate Dr. Ileana Rindasu and our Latif sir for further study. I want also to contribute and to ask question, but there is lack of time. I am not asking. Thank you very much for that this contribution of that Dr. Ileana Rindasu and our Latif sir. Thank you very much.
Thank
Next
you.
time I shall ask questions.
Yeah. So thank you. Since there is time to wind up, I thank you, Dr. Ileana Rindasu from on behalf of International Forum for Promoting Homeopathy for your time and sharing of knowledge with us. We will be definitely contacting you again regarding a discussion session on the same topic. So for the meantime, our next session to start soon. So we wind up today this session, international session, moving on to the local language session. And thank you very much for all those who participated today from different parts of the world, and especially to Dr. Ileana Rindasu from Romania. So thank you all.
Thank you so much, Christoph, for the invitation. I was very glad to participate and happy to share what I could with you.
Thank you, doctor. Thank you. See you again.
Bye.
Bye. Soon we will be starting the next session in Malayalam.